Hello. Thank you to the organizers for inviting me to come here and talk to you about some of the recent work that we've been doing looking at algal-bacterial interactions and the role that metabolites play in these types of dynamics. So I'm at Lawrence Livermore, and between when the abstract was submitted and now, I'm now a research scientist, which is great because like the bacteria to the algae in this scanning electron microscope image, I've become very attached to this project, and so this offers the opportunity to continue work on some of these interesting questions that we have. Before I start, I'd like to acknowledge collaborators on this project: Ben Stewart, Jeff Kimbrell, Peter Weber, and Xavier Mayali. We're all at Lawrence Livermore National Laboratory, just down the road. And I'd like to also acknowledge JGI for their CSP, which funded part of the work that I'll be presenting today. So just to kind of describe the image here, we're working with a diatom called Phaeodactylum tricornutum, and you can see algae, the algae with attached bacteria and flagellated bacteria. And so this is kind of the microscale scene that you can kind of keep in your mind when I'm describing what's going on. But first, kind of zooming out, why are we interested in microalgae? Microalgae are key agents in the global carbon cycle. They incorporate significant amounts of carbon dioxide from the atmosphere via photosynthesis, but they're also a potential biofuel feedstock. And so we're interested in kind of leveraging both of these pieces of knowledge to create robust and resilient algae for sustainable and renewable fuels. But the main question of this is how. And before we can kind of get to the how, you have to kind of understand your problem. And that stems from the fact that we lack tools and methods for looking at pathogens and predators within these algal biofuel systems, as well as we're missing critical components of understanding of what the bacteria within these microbiomes of the algae are actually doing and how they're affecting algal physiology. And so over the past 3 years, our question has centered on this: how do these algal-bacterial interactions shape the productivity of these energy-producing systems? And again, we're looking at Phaeodactylum tricornutum, and we're specifically focusing on the attached bacteria because we know that there's an interaction there, and we know that there's some sort of communication involved. And since this work has been started, we've since obtained approximately 24 isolated bacteria for which we have genomes, and so we're moving forward towards pinpointing how these interactions play out across different situations and conditions. Excuse me. So before I get into everything, I just want to introduce one tool that we use predominantly in our investigations, and that's the nanoscale secondary ion mass spectrometer, or NanoSIMS. And we're using this instrument to get at carbon fixation of the algae at the single cell level. So what we're doing is we're incubating with stable isotopes, we're mounting, and we're preparing the sample, and then we're ion imaging. So you can think of this as the algal cell with an attached bacteria. We're rastering an ion beam across it, which is ionizing and releasing secondary ions, which we can then split out and detect and measure the ratio of the stable to the, in this case, 13C to 12C via various detectors. So some of our previous work has shown that algae with attached bacteria exhibit enhanced carbon fixation up to 66% compared to azenic algae that have never been in contact with algae at all. And so these are in enrichment cultures containing anywhere from 40 to hundreds of individual bacterial OTUs, or ribosomal sequence variants, or in our case, it's ribosomal sequence variants. And so, as I mentioned, we've since isolated many of the bacteria within these enrichment cultures, and we've then performed NanoSIMS on those again. And what we see is a variety of algal fixation rates at the single cell level. So just to orient you, the y-axis is the incorporation of the C13 bicarbonate, and the bars are medians with interquartile ranges. And again, we see a range ranging from no effect on single cell carbon fixation to up to a 8 or 24% increase, specifically in Algoriphagus species ARW1R1 and Marinobacter species 19GW. And so I'm sure everyone's thinking, and we thought also, what role are metabolites playing in modulating these types of interactions? And we were first interested in kind of a relatively well-known metabolite called indole-3 acetic acid, which some of you may be familiar with. And it's essential metabolite in algal bacterial interactions. How it works is that tryptophan is incorporated by the algae, or by the bacteria, sorry. The bacteria uses one of several pathways to convert that into IAA. And it excretes that, and the algae incorporates it, and it upshifts cyclins within the nucleus to promote growth. The algae then release more organic matter, and the bacteria grow. So this type of mutualism is described in Amin et al. In 2015. And so we were interested in, can we measure this IAA production in these bacterial isolates by adding tryptophan and identifying what's going on there. So our experimental approach was to use these two bacterial strains that I've described. Algoriphagus is a sphingobacteria, Marinobacter is an alteromonad. And as I showed in that NanoSims data, they attach to and enhance carbon fixation. And so we were super pumped to start this work. So we started it before the genomes were finished, and the genome information came online a little bit later. But we eventually selected these two particular strains because relatives within IMG had the pathways for IA production as well as for producing the intermediates. So what we did is we grew up these cells in media and added very trace amount of C14 labeled tryptophan. We then pelleted those cells, washed them, and extracted them in cold methanol, and then sent them to the PAMS instrument. And PAMS stands for Parallel Accelerator Mass Spectrometry, Molecular Mass Spectrometry. And the way this instrument works is that we have a sample extract. It goes in through an HPLC, which is routed into a flow splitter. And in one path, it goes along to this moving wire interface, and then it goes into the actual accelerated mass spectrometer. And what this is doing is it's giving us very sensitive detection of the C14 signal coming off of these fractions that contain the metabolites. And then the other path is going into a time-of-flight mass spectrometer, where we're getting these mass-to-charge ratios to identify the metabolites. And these paths are split off at more or less the same rate, and so you can correlate the C14 signal with the actual identity coming off. And so what we see is that the tryptophan metabolism of these two strains differ. For Marinobacter, we see that basically none of that tryptophan is metabolized into recognizable metabolites. And this is shown here by the metabolite trace. It's almost all tryptophan, and that it matches the media blank that we ran. The story is quite different for Algoriphagus. What we see is that there's two major peaks that contain the C14 signal, which correspond to niacin or vitamin B3, and an intermediate known as 2-amino-3-carboxymucinate semialdehyde. And we did not notice, sorry, that's cut off. We did not notice any IAA or IAA intermediates, either in the cells, and then there were none of these tryptophan-derived metabolites in the spent media extract as well. And so by the time we kind of got this going, we got the genomes, and so we're like, okay, well maybe this is something that is genomically derived. And so we looked at the genomic potential for this indole-3 acetic acid production as determined in IMG. And so these are all of the biosynthetic pathways for indole-3 acetic acid that I mentioned. And the main takeaway that I want you to get from here is that only Algoriphagus has a complete IAA pathway through this tryptamine and indole-3 acetaldehyde route. And what this suggests, since we actually didn't measure any IAA, is that IAA production is tightly regulated. And this is in agreement with a paper by Palacios et al., where they found that IAA production was almost always undetectable unless you really pushed in a lot of tryptophan and you added an algal component to the system. So these results aren't exactly surprising, but interesting nonetheless. For niacin production in Algorphagus, we saw a nearly complete biosynthetic pathway. It was missing this one enzyme, arylformamidase, which converts L-formylkynurenine to L-kynurenine. And so what we think is happening there is there's potentially another enzyme that's catalyzing this reaction or something along those lines. And so we kind of were left scratching our heads after seeing these results, but then trying to put a positive spin on it and thinking that most of the interesting questions in science don't end in a eureka, but rather end with a huh. So our future work is going to start trying to tackle this question of what's going on between these particular bacterial strains, as well as some of the other ones in our strain library and the algae. And to do that, we're going to try to optimize an algal bacterial co-culture system. And this is really to try and get at, is niacin affecting algal growth physiology? It's currently understudied, and there's almost no papers looking at it that I could find. So it's kind of an interesting question. We also think that this might be a way to stimulate the indole-3 acetic acid production pathway in these various bacterial strains. Jeff has been doing some interesting work at identifying gene clusters and operons, and we're looking at this as a way to kind of figure out how to manipulate this system or figure out how transcriptional regulators are being turned on in response to various stimuli. And then we're also going to be working on optimizing the actual molecular mass spectrometry system, as well as the chromatographic separation to maybe pull apart those fractions that are coming off and that are being analyzed. So kind of further along in the future, we want to use this approach to eventually interrogate cross-feeding and seeing the role that plays in affecting this algal bacterial co-culture microecosystem, as well as look at the effect of co-metabolism has on production of metabolites of interest, specifically the role that maybe these algal exudates play in upshifting the potential for metabolite production. And with that, I'd like to thank friends and collaborators and funding sources, and thank you for your attention.
Any questions?